Hello everyone. So in this session we are going to study about the tRNA, the adapter molecule. So in the last session we were studying about the salient features of genetic code, and with that we also studied about the point mutation and frame shift mutation. Point mutation is change occurs in only one base pair, and the frame shift mutation is change occurs in 2 or 3 base pairs. So that leads to diseases or disorders. So after that, after all those, we are going to study about the tRNA. The T represents, or T stands for transfer. Okay, so T stands for transfer RNA. Actually, our process is we should about to study. We should we have to study about the what is that translation process. We should be in translation process, but before going to start the translation process, translation is nothing but the mRNA, it synthesizes the protein molecule. That is a simple form of definition of translation, but actual process, the actual definition I will tell you when I start the translation process. So to understand the translation process, we have some what is that, some preparation, some key points to remember or to understand better. If we understand all these, what I am going to tell now, if we understand these concepts, it will become very easy to know the mechanism of translation. So the first thing I would like to start here is the tRNA. About the tRNA, it is an adapter molecule. Why it is known as adapter molecule? Adapter molecule is it is something, it is adapting. What actually it is adapting? Still we doesn't know. We didn't learn the structure of tRNA. T stands for transfer. Something it is transferring. We have to understand that, right? So the answer is, I'll start by drawing one diagram so that we come to know what actually it is transferring. So now we are having a structure called as tRNA, which looks like clover leaf. Okay, so here this end is having the meiotic 5 prime, and this end having the meiotic 3 prime. This is 5 prime and 3 prime. The arm which is nearer to the 5 prime is known as DIHU loop. So DIHU stands for dihydrouracil. Dihydrouracil loop. And here one more arm is there, no? So this arm refers to anticodon site or anticodon loop, whatever you can call this arm. And here one more arm is there which is nearer to 3 prime. So this arm is known as pseudo-thymine or thymine loop. So which structure we are studying? Student, just concentrate here. Which structure we are studying here? We are studying the type of RNA, right? So in the RNAs, the nitrogen bases, what type of nitrogen bases are present in the RNA? One is adenine, guanine, cytosine and uracil. But here, if you observe this, one thymine loop is present. That thymine loop consists of a pseudothymine nucleotide. Okay, so the sign represents, so the pseudothymine can represent by writing one sign like this, writing U and putting one dash. So in the next concept, so writing this loop after this loop, what I told you now, one thymine loop, and it is known as pseudo. Before that I wrote pseudothymine. That is not correct. Just make it correct. This word, this symbol is known as pseudouridine. It's not pseudothymine, but the loop is known as thymine loop. Okay, but in the NCERT book the structure is little far differ from this structure, that the structure remains same, but what they have done, they have drawn the structure as it is, how it looks in the cell, the structure as it is, same content they have given in the NCERT book, but here the actual shape I have drawn here on the board. Alright. So next is at the 3 prime, it is having CCA codons, triplet codons, and this site here it is having a 3 prime end, means there will be OH group. The OH group is the functional group. So listen to me carefully. So this end is having the moiety 3 prime. Always at the 3 prime the OH end will be free. The OH end or the OH group is the functional group. So to this functional group, amino acids comes and attaches to this site. So this the amino acids after getting charge, they'll come and they get binded to this site, to this end. So this end is carrying the charged amino acid. So one more end, one more loop we are having, one more arm we are having, that is dihydrouracil. What is the function of dihydrouracil? This uracil loop or a DIHU loop, it recognizes. What it is recognizes? It recognizes it helps to recognition of amino acid. So what is the use of recognition of amino acid? You'll come to know when I start the translation process. You'll come to know recognition of amino acid. So this is what I have written here is the function of this arm. And at this site amino acid is going to bind, and here the site is known as anticodon site or anticodon loop, we can call it as. What is the function of anticodon? This anticodon, it will trace the nucleotide sequence which is present on the mRNA. Consider the mRNA. What we have drawn here, this mRNA, remember always RNAs are single stranded. It looks like double stranded, but it is a single stranded molecule, single stranded RNA. This is also a single stranded RNA. So consider, just suppose here we are having an mRNA strand. On the mRNA, it will be having the triplet codons. To make you understand, I am just writing a few nucleotide sequences, like a few triplet codons like this. like this. All right. When it start the process of translation, what it will do, it has to read the triplet codon sequences. It has to read which is going to read the nucleotide sequences, the tRNA. Particularly this arm, this end will read the triplet codons, the sequence of nucleotide. Consider only these three, this triplet codon we are going to trace. So here it is having UCA type of triplet codon. The UCA type of triplet codon, the anticodon site will be trace the opposite sequences. The anticodon site will trace the opposite sequences, like it will code the opposite nucleotide sequence of uracil is A, for C G, for A U. Like this it will code the tRNA anticodon site will code for this with this mRNA. All right. So this is how it keep on reading the mRNA nucleotide sequences. Just understand in the simple way what you have to understand here. It will take the opposite nucleotide sequences. It will read, it is like in the previous classes we understood about the DNA strands, double helical model of DNA. Here if it is having adenine, the opposite strand should be T, right? So A always bind with the thiamine molecule, like this. Always uracil binds with adenine, cytosine binds with guanine, adenine, cytosine, adenine always bind with uracil. Now it get link, right? So here if you observe this, AUG triplet codon is present at this site, and here one more triplet codon is there, here one more, here, number of sequences will be there on the mRNA strand. In the last, based on the salient features of genetic code, AUG acts as initiator codon. Actually the process should begin from this site, right? So to start the process, one more tRNA is there, known as initiator tRNA. So that initiator tRNA will be having the triplet codon like UGC that can bind with this AUG nucleotide sequences or AUG triplet codons. How many tRNAs are there now? One is initiator tRNA, one is the actual process. The when I start the translation, you will come to know. Just remember here, to start the translation it need two tRNAs. One will be the initiator tRNA and then one will be the subsequent, following tRNAs. Then how many tRNAs will be there? Number of tRNAs will be there, number of ribosome rRNAs will be there, mRNA strands will be there. So tRNA is, one is initiator tRNA. After binding to this AUG, after taking one amino acid, remember one more thing, always AUG code for methionine amino acid. If it is AUG triplet codon, it should bind with the amino acid methionine. Check it out in your checkerboard, genetic code checkerboard. AUG codes for methionine. In the last class also we understood the AUG triplet codon has dual function. It is an initiator codon and also it binds to the methionine amino acid. So later on, don't get confused. Still I have not started the actual process. Only to make you understand, I just wrote 2 tRNAs. Okay, so when I start telling the tRNA process, you will come to know why it requires 2 tRNAs. Okay, so what we understood here, so this arm is known as anticodon arm and this side is going to copy the triplet codon sequences from the mRNA. One more arm is there at this side, that is known as thymine loop or thymine arm and pseudouridine. The function of T loop, also known as T loop or thymine loop or pseudouridine loop. So T loop or thymine loop, the function is it recognizes the ribosomal RNA. Ribosomes. It recognizes the ribosomes. And the next is here, what we are having here at this end, the 3' moiety. What it is going to bind? It is going to bind with the amino acid. This loop is recognizing the right amino acid and this loop is recognizing the ribosomes and this is going to code with the mRNA. So after all this, what is the use of understanding the T loop, tRNA to start the process, to initiate the process? So the process will go in 3 steps like initiation, elongation and termination. As like transcription, the translation is also follows the same, but here in the translation process it is little bit having some extra concepts. So now according to your NCERT, I am going to draw one diagram to make you understand how that resembles the tRNA. Yes, dear students, we have the structure according to your NCERT. So this is one tRNA, one more tRNA. So here they have given this codon and this is the mRNA strand. This is the mRNA strand. Number of triplet codons will be there. Only they have selected 2 random triplet codon, right? And this is one codon. So this codon, to this codon, the UCA anticodon comes and binds like this. And remember, the AGU triplet codon binds with the serine amino acid. If it has on the mRNA strand, if it has AGU triplet codon, the tRNA brings the serine amino acid with the at the 3' end. So why? Because the 3' end has one OH functional group that brings the amino acid, or vice versa. The activated amino acid comes and attaches to this site, or the tRNA brings the amino acid, however you can understand. So here to the AGU codon, triplet codon, the serine, the triplet codon codes for serine amino acid, right? So they have given like this. This end is known as 5 prime end, and this end is known as 3 prime end, and they have not mentioned any loops here. That's why I draw that diagram. And so this is only one strand. Remember, single stranded tRNA it is. Because of the nitrogen bases get attached between the single strand, it looks like double strand. And then what next? Here one more codon they have given in the NCERT book. The UAC codon codes for tyrosine amino acid. Okay, have you understood this? To code for only to get attached to this UAC, the anticodon end, the anticodon arm, it will like it will have the complementary base pair like AUG. All right. So this is what about the tRNA. Once in the last year they have given one question like tRNA is an adapter molecule. Comment. For 5 marks you have to understand, you have to answer that for the question. So what you will write? So you have to write like the tRNA or adapter molecule. It has the binding site, amino acid binding site at the 3 prime end, and one more arm, one more loop is there. So that loop or that site codes with the triplet codon which is present on the mRNA. And one more loop it is having, it will recognize the amino acid, and this arm recognizes the ribosomes. All right. So the tRNA, it is playing many roles. One arm is attaching with the amino acid, one arm is coding with the mRNA, one more arm is recognizing the ribosomes. What is the role of ribosomes here? So that I'll tell you. One arm is recognizing the amino acid, that's why it is known as adapter molecule. And one more thing you have to understand here. So this tRNA, it is transferring the amino acids. So what is our aim? We have to understand how the protein molecule synthesizes. Consider, I'll write a simple protein chain I'll write here this is one, consider this is one amino acid, one more amino acid, one more, like this a number of amino acids, they get linked with the bond known as peptide bond, right. So this peptide bond should form here. We have amino acid, we have the nucleotide base pairs. What is left over now? The leftover thing is the peptide bond should form in between one amino acid and one more amino acid. How it will form? Where is the site of forming the peptide bond? How the peptide bond synthesizes, or one amino acid and one more amino acid, how they link? So that will understand. Just remember here, we are going to understand the protein, synthesis of protein. This is how our structure should be. Okay. So the next is, let me tell you a simple form or how the transcription and translation process occurs in a cell. Consider a cell, right. It is having nucleus. Inside the nucleus, the DNA synthesizes, right. So after that the DNA synthesizes the RNA, hnRNA first and then next mRNA. After the process of splicing the RNA, it will come out from the nucleus. Don't write this diagram. Okay, so just understand where the processes are going on. The transcription process is taking place in the nucleus, correct? After the transcription process, after the splicing and all, the mRNA, it is coming out from the nucleus, correct? So this nucleus now, after this mRNA strand here, it has to code with the tRNA, and it has to recognize the ribosomes and all. Where are the amino acids, where are the tRNAs, where are the ribosomes? So the ribosomes, if you have remember, in the class 11 we studied about the cell, the unit of life. While studying the cell, we come across the cell organelles, right? So while doing the cell organelle, while doing the endoplasmic reticulum, I mentioned one word known as smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum is nothing but having ribosomes on the surface of the endoplasmic reticulum. Yes, those ribosomes. So those ribosomes, suppose ribosomes are present on the endoplasmic reticulum. Now our mRNA strand is ready now. What left? Amino acids and tRNAs, right? So first of all, to start the translation process, transcription is done now, ribosomes are ready, amino acids are also present in the cell, and tRNAs also, they will be floating in the cytoplasm. Now the mRNA, it came out from the nucleus, the matured mRNA, the mature RNA. The first thing, the amino acid, it will get charged. How it will get charged? By taking the ATP, the energy, the amino acid, they will charge first. Later on, it will come and bind with the tRNA. The tRNA having a charged amino acid, that tRNA is known as charging tRNA or amino acylation tRNA. That tRNA, now what it should do, it has to come nearer to the, or at the mRNA. It has to come near to the mRNA. It will come, no problem. Before that, for the elongation process, we need the ribosomes to initiate, and for the elongation we need one more molecule, that is ribosomes. The ribosomes, they are of two subunits. One is smaller subunit and one more is larger subunit. To the smaller subunit, consider this is the ribosome, structure of ribosome, and this is smaller subunit and larger subunit. Okay, at the smaller subunit, the mRNA, this mRNA, it get bind, it get attaches. The mRNA attaches at the site of, or at the smaller subunit of ribosomes. Where are the ribosomes? Ribosomes will be there on the endoplasmic reticulum itself. The mRNA goes to the ribosome and it get attaches to at the smaller subunit. And then next, the larger subunit also, it get attached to the smaller subunit. Larger and smaller, and mRNA is ready now. What left? The charged tRNA. that charged tRNA, this will bring the amino acids now. The amino acids, it need 2 tRNAs. Okay, one tRNA will attach at the initiator codon, one more tRNA will attach next to that, and then what will, so this is the initiation process. What will happen next? It should move forward. So when it start move forward, when the ribosomes start moving on the mRNA, the first tRNA, what it get attached at this side, it get detached. So this will take this place, and one more tRNA comes and attaches to this side like this. So consider this is the mRNA, one tRNA is there, one tRNA is there. When the process starts, the first tRNA get detached, and then this tRNA again it will move little bit forward. One more tRNA which is floating in the cytoplasm, it will bring the charged amino acid. So like this the process, it will keep on moving. Only I have described briefly. This is only the translation process, okay, that takes place in the cytoplasm. Still I have not started the process of translation. I have to start now, after understanding this, we will understand the translation process in the next class. Okay, so if you have any doubts, please ask me. Thank you.